Well, kia ora tātou again, everyone, and welcome to this webinar. It's lovely to have you here with us. We'll get underway because people can still join us if they're just a little delayed. But we want to stick to the golden hour and give Professor Justin as much time as possible to speak about this topic. So, is Parkinson's genetic? And I must confess that this is something that I want to know because my dad had Parkinson's. He was diagnosed at the age of 70, and I was diagnosed at the age of 55. So my siblings are all much older than me, and none of them have got it. And just the other day, I heard for the first time of 3 generations in a family who've got Parkinson's. And so is this Parkinson's genetic? And we're just so privileged to have someone of the caliber of Justin O'Sullivan working. He's the head of the Liggins Institute at Auckland University. But working in this area of genetics and the research. And so we're so fortunate to have a world expert to address us and try and answer that tricky question. So Justin, thank you so much for your time and your expertise, and we hand over to you now.
Thank you. Now I'll just get it set so I can see. Right, sorry, I'll get rid of this control bar. Cool. So thank you very much for having me here today, and thank you for the very warm welcome, Andrew. The question, is Parkinson's genetic? Is an obvious question to ask, and the real simple answer is that there is a genetic component to Parkinson's. And as I go through my talk today, I'm going to try and tell you a little bit about the stuff we've been doing and how that links into that, and so how we're trying to work with what we understand the genetic component to Parkinson's is. It's important to understand that there are different forms of genetics that might impact on Parkinson's, and there's a certain amount of risk that's associated with genotype. So it's not necessarily that your genes mean you will get Parkinson's. It's not definitive or deterministic, if you like. Instead, really what it is it's about risk. But there are some forms of Parkinson's where there are familial genes. There are mutations in genes like SNCA, like LRRK2, and other genes where basically the risk of Parkinson's is much, much greater, or the onset is much earlier. And so there is definitely a genetic contribution to Parkinson's, but it's also environmental. So there's an environmental interaction. Your genes and your environments really do interact. And it's that interaction which manifests the risks that we have that are inherent to our genetics. And so as we go through the talk today, that's really what I'd like to stress, and I'll talk to you through what we understand at the moment. So let's start with Parkinson's itself. What is it we're talking about? And I know talking to you that this is probably, something you're all aware of, but Parkinson's disease is a chronic and progressive movement disorder. It's a physical manifestation of the toxic alpha-synuclein aggregates and the resulting death of the dopaminergic neurons that occur within the central nervous system. That is a great definition for somebody like me. It's maybe not the best definition for other people. So let's just break this down a little bit, okay? So as you were diagnosed, many of you will have had issues with movement and things. You went to a doctor and went to a specialist, probably through quite a long journey, unfortunately, to get there. But you went through this process and you were diagnosed with Parkinson's as a result of that process. The physical manifestation of Parkinson's occurs basically when about 70% of the dopaminergic neurons in your substantia nigra have been destroyed by protein aggregates that seem to form and affect those cells. And those aggregates, it's really simple to think about. So this little sticky ball that I have here, many of you have seen these. These are things that you get and throw at things, children's toys, right? They have a little Velcro on them here around the ball, and this one has a tail on it, and the tail's here for a specific reason I'll explain later. But this ball you can think of as the alpha-synuclein protein. And basically what happens is that one there, that protein sticks to this one, and it sticks to another one, and it forms an aggregate of proteins that really come together, right? And so this aggregation of the proteins is bad. And it's particularly an aggregation of alpha-synuclein. Now, similar things happen in other conditions like Alzheimer's, where you get an aggregation of another protein, and that amyloid plaque forms, and then that amyloid plaque destroys or aids the destruction of the cells around it. And that gives you what is associated with Alzheimer's. And there are other forms where you might have heard of, like a prion for mad cow's disease. And that prion forms aggregates, and again, that's associated with the destruction of nerve cells and brain tissue. And that forms a particular condition. Different genes, different proteins, but they are all resulting in a similar thing, which is ultimately the formation of these sort of protein aggregates, which are then seen to be affecting and destroying the nerves and the cells around them, or contributing to that. And so it's the resulting death of the dopaminergic neurons, which really happens in Parkinson's, and that's associated with problems in the nervous system, and that's what the manifestation is, a pathological manifestation of Parkinson's disease as we know it. The problem is that all of you are different. I mean, it's not a problem, it's a reality. You're all very different. In fact, Parkinson's disease is an umbrella term which encompasses a lot of different subtypes. No two of you have the same form of Parkinson's disease if you have it, or no two Parkinson's patients have the exact same form of Parkinson's disease. And that's really the key, because what happens in the condition, as many of you will know, is you develop different comorbidities or different manifestations of the condition. And this is really very important. And it's important because if we think about it, we classify diseases by pathology. And in the description that I just gave you, we talk about this, that the manifestation of Parkinson's really, when you end up going to the doctor and the clinician, is really at that point about 70% of your neurons, your dopaminergic neurons, have been destroyed. And that's what's affecting movement, and that's what's affecting those characteristics that you have. It's really simply. But that is really late in the process. It's really late in the process of developing the condition. And that's a pathological manifestation of the condition, Parkinson's. Now, Parkinson's is defined by that because it was first defined originally, its formal definition was way back in 1817. And its definition was done by James Parkinson, and he did it in 1817, and it was about movement, and it was about the way people moved and behaved. And since then, there are a lot of conditions that have also been defined in a very similar way. So things like autism, things like Huntington's disease, things like schizophrenia, all of these conditions have been defined in the late or early 1800s through to the early 1900s. And so that's great. We've known about these conditions for a long time, but these definitions are all defined on pathology. And this is a problem for us because in many ways, and in some ways, those definitions haven't moved very much since then. But if you think about it, what's actually happened since sort of 1800s in terms of our technology and everything else. So to put that into context, in 1896, Marconi got the patent for the radio. Over the subsequent years, we've moved from really, really basic radio communication right through to what we're doing right now at this minute, where we are communicating across computer network, and it's a communication between, over 50 people instantly, to the point where you can ask questions by typing them into your computer, right? I mean, the difference in what we're talking about here is massive. The technological changes that have occurred is huge. And yet the way we think about Parkinson's as a condition hasn't really changed a huge amount since 1817. And that's really a problem. So, do these descriptions help us? Well, in some ways, what we're doing when we're defining people for Parkinson's is we're putting people into one box, right? We're saying Parkinson's, this is Parkinson's, you have Parkinson's, here you are. And we're trying to squeeze everybody into this single box. But we know, as I said, that everybody's different. We know that everybody's Parkinson's manifests in slightly different ways, that this comorbidities or the conditions that co-occur with Parkinson's are different, that when they occur are different. So categorizing the disease and categorizing Parkinson's like this helps us really, categorizing the disease and categorizing Parkinson's like this helps us really, helps to keep us alive in some ways, but it's also a bit of a hindrance to our living. And so this is a really important thing, because really the way we should think about Parkinson's, should shift. And that's because by the time the pathology comes through and it's bad enough for you to present, what we're doing is we're really acting as an ambulance at the bottom of the cliff. That's because we're thinking about pathology. But let's shift our thinking a little bit. And let's think about Parkinson's in terms of how does it develop. Now, if we consider how it develops, the main things that impact how something develops, or one of our traits develops, are our genetics, our genes, our DNA, and the environment that we're in. So can we get at that information? Can we understand the genetics? Can we understand how that might contribute to Parkinson's? And that's really the core of the talk today. So as I've kept reiterating, Parkinson's patients are unique. Each has their own disease trajectory, their own comorbidities. These are the sort of manifestations, other manifestations that occur with Parkinson's, whether that's gut problems, whether that's lack of cognitive or mental acuity, and things like that, right? These all change. And so your trajectory and the trajectory of the development of your condition can be thought of in terms of, oh, let me just turn this on. Sorry, I haven't turned that on very well, have I? I should have the laser pointer on. Sorry about that. Let's talk about it in terms of this mountain range. So if we think of the peaks on this mountain range as being Parkinson's and different forms of Parkinson's disease, then there are N peaks in this mountain range. Now, each individual starts effectively at a different base camp, okay, here. And the trajectory that that individual takes on the different paths that go up to this form of Parkinson's here, or this form of Parkinson's here, are different. And those paths are affected by the genetics of the individual. And this includes, of course, the gender, because gender is, at least from a biological point of view, gender is defined by your genetics. From a social point of view, it's not, but from a biological point of view, it is. It's affected by your age, by your comorbidities, so the other conditions that you have. So not just your predisposition or your risk of developing Parkinson's, but the other conditions you can have a risk of developing. And by your environmental exposures or your exposome. So what we're exposed to, whether it's things like pesticides or high levels of plastics in the water that we drink, or whatever that might be. And these things really affect the way that we go and different individuals develop Parkinson's. And you see that here. Now, each individual starts at a different base camp. You start at a different base camp, and that's defined largely by the genetics, the genes, the DNA that you inherit from your parents. And what is, of course, the biggest gamble of our lives, right? Which is when we get all that DNA and we mix and match it from mum and dad, and then we see if it's a working system. And so that gamble that we all undertake sets our position across here, and then our environmental exposure and the other comorbidities that we have, they impact on whether or not we go up here and whether or not any risk that we have for developing Parkinson's, which is genetic, whether that risk is actually manifest in the development of the condition or not over the course of our lifetime. So this is the way that we're thinking about this, and we're thinking about the genetics and how genetics might contribute to Parkinson's and to our risk of developing Parkinson's as a condition. So that's a nice thing to think about, but how can we study this? Well, scientific studies, often they come across as being very complicated, but in reality, they're not that difficult, many of them. And so there are scientific studies that can connect changes in your genes to your characteristics, okay? And these studies are called genome-wide association studies, or GWAS for short. Now, the term doesn't matter, it's just something that we use. But the key point is that what we can do is we can take a population of people, and we have a population of people here, and we can split that population of people into two groups. And we can do it by whatever characteristics we want to do it by. Oops, wrong button. So we split them into this group, and this group is the control group, and we have them right here. And we have this group here who are wearing shawls, and the shawl is the characteristic that we're looking at. Now, you can replace shawl for Parkinson's, or you can replace it for autism or any other condition you want. But in the context of today, it's Parkinson's, all right? So Parkinson's in this instance is shown by the presence of this brown shawl on these individuals, okay? So what do we do then? We've got two groups of people. Those groups are made up of individuals. But what we can do is with the technology we have, we can now sequence those individuals in those two groups, all of them. Now we can do it in different ways. We can sequence all of their DNA, which is a bit expensive, so we don't tend to do that. Or we can sequence a subset of their DNA, which is what we tend to do. So we sequence a subset of their DNA, which statistically gives us a picture of all of their DNA. So we sequence that subset, and we look at the characteristics within that subset of DNA. And when we do this, there are locations in the genome. We get sequences. So from these individuals here, we get these 5 DNA sequences. They all align. And at this point here, you see we have mostly A's, a T, and an A, okay? And that's the sequence at that point. So adenines and thymine. And that's the sequence in the control group. Now, because of the way we do this, we use the same technology on both groups, of course, means that we can compare the DNA that we get from that group here, the controls, with the DNA that we get from the people who have Parkinson's. And if we do that, what we see is we line this up. And when we line it up, we see everything matches except at this point here, okay? And at this point here, what we're seeing in the Parkinson's patients is we're seeing a predominance of the T and not the A. Now, we call this a single nucleotide polymorphism, or a single nucleotide variant, but it's more often than not, you'll hear it referred to as a SNP, okay? And that's kind of nice because it implies it's like scissors and you've cut and you've just changed one thing. But this single nucleotide polymorphism, or this change in the DNA here at this one position, is associated with Parkinson's. It doesn't occur at high frequency in the control group, but in the Parkinson's group here, you see we've gone A basically to T, all right? And that means that what we would say is that that base, that genetic base, is associated with Parkinson's disease. And that's how this assay works. And it's a very, very simple assay. It's a very nice assay. You can run it across a lot of people. And in fact, it's been run across millions of people, okay? And we've looked at this, and people have looked at this, not us, but other people have looked at this with respect to Parkinson's disease. So your question, and the ultimate answer to your question, is Parkinson's genetic, is answered by this very assay. And so the biggest one that has been done to date, and there's a bigger one being done by a project which is called GP2, but the one that's being done at the moment, or that was published at the moment, was published by Niles and people from the NIH. And they basically looked and they identified novel risk loci and causal insights for the heritable risk of Parkinson's disease. And they ran one of these assays. So the groups that they separated here was control people, and they had 1.4 million of them. And they had people with Parkinson's, and they had about 50,000 of them. And they compared the 50,000 with the 1 million, the 1.4 million, and they asked where in their genomes do their DNAs differ, and which ones of those changes are associated with Parkinson's disease. And when they did this, they found this. They found that there were 90 changes, 90, that's all, at 78 different places in the DNA. Okay, so a locus is just a region of DNA. That's how we define it, as a part of a chromosome. But 90 changes in 78 places in the DNA. Now let's put that into context for a second. So 90 changes. 90 changes in how many? Well, your DNA it consists of approximately 3 billion base pairs. So 90 single changes in 3 billion were associated with the risk of Parkinson's disease in this cohort of people. That's all. Very small number, but those 90 changes explained up to 36% of the heritable risk of developing Parkinson's disease. Okay, so if you do a twin study, you can work out the heritable risk of developing Parkinson's. Well, these variants explain up to 36% of that risk. Now, that's quite a lot of risk. Obviously, there's another contribution, which is the environmental component. But these 90 genetic changes can contribute to your risk of developing Parkinson's and then interact with the environment. And that is the full manifestation of risk, at least as we understand it at the minute. Now, this is pretty powerful, right? If we know there are only 90 changes, it should be pretty simple to understand what they're doing, right? I mean, 90 changes in 3 billion, we're down already orders of magnitude. We should be able to study that and understand that very, very quickly. You would think so. The problem is that we tend to think about DNA in terms of what are present, or units that are present within it, which we call genes. And those units, those genes, they encode all of the proteins and the enzymes that make up our cells, that make us work and function, right? And those genes are how we tend to understand things. And that's a historical understanding. But they really are the functional units that we talk about inside the DNA. Now, the problem with this is quite simple. When you go through these 90 variants, none of them actually fall in genes. All of them fall in the bulk of your DNA, which is actually what we call the non-coding DNA. Now, it was once referred to as junk. Now, people don't think about that. That was a flippant comment, actually, originally. But it's the non-coding part of the DNA. It's the part that doesn't have the genes in it as we understand genes. And that makes it very hard to understand what these 90 changes are actually doing. But there are ways. And so our group has worked on that, and Sophie Farrow, who many of you might know, have heard of before, she's done a lot of work on that with us, and we've worked around that. But I'm not going to talk about that today. What I want to talk about a little bit more is how we can move beyond that type of understanding to something that moves from an associative understanding to a causal understanding. And there's a slight difference here, okay, between something that's associated with Parkinson's or something that's causal for Parkinson's. And the difference is that associated things we don't know directly cause Parkinson's, but we can do tests that actually prove or indicate if something actually causes Parkinson's. Now, that gives us further insights into the condition and potential ways to understand how we might interfere or with the development of the condition, or in fact, look at therapeutic approaches to a condition. But to understand causality, we have to understand this guy. So this guy is Gregor Mendel, okay? And so Gregor Mendel, he was a monk, and he did the experiments that basically defined the field of genetics. He did those in the late 1800s. They were forgotten about for years, as we tend to forget about things, and then they were brought back and they were understood in greater detail and really set the scene for genetics. And the experiments that he did were really, really cool. So he bred peas, and peas, you know, if you grow them in your backyard, you'll know that you can get round ones, you can get wrinkled ones, you can have yellow seeds, you can have green seeds, you can have purple flowers, you can have white flowers, you can have an inflated pod, you can have a constricted pod where the peas are all very obvious. You've all seen all of this if you buy sweet peas in the supermarket, right? Very, very nice. Well, he realized that this was telling us something about the way the traits were passed from one plant to the other when he bred them, because he crossbred the plants. Now, when Gregor did this, he understood the rules and he developed our understanding of the rules. And those rules are really quite simple. They're simple, the rules of inheritance. And they're so simple that we teach them to our kids at school, right? And we teach our kids about Punnett squares. And the way these squares work is very simple. We say we have a gene in dad, which is a big B. We have a little b gene too, because dad is a heterozygote. So he has a big B and a little b gene, because we have 2 copies of most genes. We inherit 1 copy from our dad and 1 copy from our mum. And that's what Gregor figured out. Now, mum, of course, has a big B and a little b. So we can inherit 2 big Bs, and we do that 25% of the time. In other words, 1/4 of this graph. Or we can inherit a big B, but the big B here comes from dad, or a big B from mum. And we can inherit a little b from mum and a little b from dad. And that happens roughly 50% of the time, right? 2 out of the 4 squares. Or we can inherit 2 little b's. And the 2 little b's mean that we're getting this thing here. Now, We talk about this in terms of dominance, in terms of recessiveness, and those types of things, but it's true for any two genes. And so we teach this to our kids because these rules are simple. It's a nice way to understand how traits are passed, how things come to us from our parents, and how we pass things on as part of our DNA and as part of the whakapapa that we pass on to our children. Simple rules. Except that we tend to sometimes pretend that things are simple, and they are in this instance, but they aren't. And the classic example of that is eye color. Okay, so eye color is generally taught as a Mendelian trait. You know that there's a brown gene, there's a green gene, and those genes are what we call codominant, which means if you inherit those, you display traits of both green and brown eyes mixed together, which I think is hazel, isn't it? Something like that. But you also get a blue, and the blue is recessive. And so you can have a brown and a blue, and then your eyes will be brown, or you can have a green and a blue, and your eyes will be green, or you can have two blues, and then your eyes will be blue. But your parents could have brown eyes, and you could be blue-eyed because they both have recessive blue, and they pass that to you just through that Punnett square. Sounds simple, except there's actually 236 degrees of freedom in eye color. There are all sorts of patterns. There's get a hazel coating around your eye or a shell around your eye, all these sorts of things. And the reason that works is basically because, again, with eye color, a lot of the signal comes from these pieces of DNA that are outside of genes, not the genes themselves, just like it does for Parkinson's. And so those pieces are really important to understand. So that means that the rules aren't quite so simple, but we do understand a lot about them. And the cool thing about Mendel's rules are that they actually help us define a statistical method. And the statistical method is called Mendelian randomization. And so what Mendelian randomization does, and this is the technical term for it, I'll just run that past you really quickly and then I'll explain what it is it's a complementary approach that uses natural occurring genetic variation to identify possible causal effects between a risk factor and an outcome in a time-efficient and low-cost manner. Okay, great phrase. You're all sitting there going, what does that mean? Very simply, what it means is that we can use people as if they're a natural experiment because everybody has variation. And if we use the variation that people have, very similar to the GWAS study, but in a slightly different way, we can connect that through to the trait, in this case, Parkinson's, but in a way that actually allows us to infer causality for the gene that is being modified and not simply for the activity of a gene that's being modified and not simply for the genetic variant that occurs outside of the DNA, outside of the genes in the DNA. And so this is the key thing. It uses variation that naturally occurs in people as part of a big experiment. And that big experiment allows us to say that something is potentially causal for Parkinson's. So let me show you and explain this in a simpler or a slightly different way. These are forks. You've all seen them. Many of you will have used many of them. Some of you might have used all of them. Some of you might have only used a few. I've only used a few. But, we've got different ones here. We've got this fork. It's an oyster fork, 3 tines. It's a short fork, bit stubby. It's good for picking up oysters. They don't slip off it. This one is a cheese fork. It's got a good handle on it so you can grab it. You can really stick it into the cheese. Got 2 nice long prongs so that you can go into, or 2 long tines so that you can go into a little deep block of cheese and you've got a good picking it up, etc. This one's a pastry fork. It has almost a blade on this edge of the tine here, which is variant, which allows you to cut the pastry. Spaghetti fork, obviously the outer tines have got these things, which means you can wrap the spaghetti around it as you're digging it in and pulling it out. This is a crab fork. Looks really different to the rest, right? It's designed so that you can actually pull the crab meat out of the legs, right? Narrow extension here, 2 short tines, dig it in, pull the meat out. We could go through the rest of them. We won't. But these are the variations in forks, right? Now, obviously, each bit of those variation affects the activity of the fork. They all do the same thing. They're all to pick up something, right? But if you try and eat spaghetti with a crab fork, you're going to have an awfully bad time of it, right? You might get 1 strand of spaghetti. You might be able to twist that between the tines, but that's all you're going to get, right? And likewise, if you try and eat crab with a spaghetti fork, it's not going to work very well either, right? You might saw through the bones or something, or the shell, but you're not going to get much crab, So the variation is important for the functions of the forks, for their traits, okay? And so let's think about this here. Let's explain. If I have a set of forks and I go into it, there's some paper forks here, these ones. They've obviously replaced the plastic forks that we used to use, which were very bad for us or our environment. But these paper forks here we have. Now, those paper forks, when I go and I get them and I pull them out, generally I'm thinking of this. I'm going to go to the beach, I'm going to have a barbecue, and I'm going to enjoy that and have that. The thing is that whenever I get these forks and I pull them together and I start to do that and prepare to go to the beach, what actually happens is this: it rains without fail. In my experience, okay, connecting these forks, those forks cause bad weather. Now, it's a bad example, right? I'm exaggerating because we all know that forks don't cause the weather. That's all done by butterflies flapping their wings in Mexico. But realistically, the point here is that these forks, in my experience, are causal for bad weather because I can never get to the beach when I select. Now, Mendelian randomization does just that, except the example is not silly. The example is that it's actually linked to that condition. Now, we can apply Mendelian randomization to actually understand Parkinson's disease and how it forms. And so we've done that in a particular way, in a very nice way, in a way I think that we've published, and we published this recently. It was published by Shrimo here and Tayaza and Sophie and Anthony, who's at the Garvin Institute with us. But we published this paper earlier on. So if you click on that little thing there, or you take a photo of that, that will take you to the paper. But the approach basically connects our understanding of DNA variation, the SNPs that we were talking about, those single nucleotide variants that occur throughout your genome, with the epigenome. And the epigenome is, in this instance, is the structure of the DNA and the methylation and things that occur on the DNA. That's really the signals that come from the environment and modify how your DNA is read and acted on. It combines DNA variation and an epigenome with gene regulation. That's how we turn genes on and off, in combination then with studies into the genetics of Parkinson's disease. And in doing this, it really allows us to look in depth at what might be causing Parkinson's disease itself and the genetics of that. Excuse me. So when we do this, what we find is this. Okay, we did this and we ran this on the brain. So we made up a special network of genes and gene switches in the brain and how they all work. And we ran the Mendelian randomization on that against a big cohort of people that have Parkinson's, and we asked which genetic variants and genes are linked to Parkinson's in the brain. And when we ran that, what we found is this. Okay, so very, very simply, the ones on the left here, to the left of 1, anything on this side is protective against Parkinson's disease, right? So if you have this particular genetic variant, which is determined by this number here, specified by that number, it doesn't matter what it is, and you have this gene, which you will, then if you have that particular combination, then you have greater protection against Parkinson's disease or occurring Parkinson's disease. So in this example here, this particular one gives us about a 40% protection against Parkinson's compared to somebody, a control, who has a risk of 1. By contrast, these genes here, in particular STX4, gives us an increase here, a 50% increase in the risk of developing Parkinson's. So if you have this particular genetic variant in combination with that gene in the brain cortex, then basically your risk of Parkinson's goes up 50% of developing Parkinson's compared to a control person, okay? And that's what this is all about. It's about risk. But these genes here, and many of them are known or have been associated with Parkinson's for before, like RGAT27, STX4, CANZEL1, LINK2210, they've all been associated with Parkinson's. They affect your risk of developing Parkinson's. So the switches that turn them on, and these are the switches, in combination with those genes, affect your risk of developing Parkinson's disease. Either protection or risk, okay, increased risk. But this information actually allows us to do a lot more than just that, right? What we were able to do here when we did this was we were actually able to take this information, and because we know that genes and proteins don't work by themselves, they work in big networks, we were able to use that information and try to understand more about the actual pathways that are affected in the brain and get insights into things that might actually affect those pathways and might actually be useful for therapeutics or other approaches to study Parkinson's. And when we did this, I'll just show you one example. This is one example of what we found. And so this is an approach, and it gives us insights into potential treatments. Okay, and these are just potential. The gene that we found that was causal for Parkinson's was CD38. Okay, it's a cell division control gene. What it does is a cyclin. It doesn't matter. I won't go into that in too much detail. But this gene here is part of a pathway in the brain. It interacts with these other genes here, NMNAT1 and NMNAT2, and this one here, which is called NADSIN1. Names are terrible. We call these things horrible names. But the point here is that these genes interact. These genes in the brain are part of this pathway, which is called the Priest-Handler pathway. And the Priest-Handler pathway is all to do with the generation of NAD. And NAD is a really important energy source and source of electrons in cells, okay? And so this pathway is all to do with the generation of NAD, we think, in the brain, where we identified it. Now, the interesting thing about this is that this NADSIN1 protein here, which isn't causal, but is part of this interaction network that we saw here, is regulated by these genetic switches, these single nucleotide changes that are in your DNA. Now, they all regulate NADSIN1, but what they also regulate, interestingly, is vitamin D synthesis. And they regulate vitamin D synthesis in the skin. Now, that's not got anything to do with NAR or NAD, but to do with vitamin D. One of the interesting things about vitamin D is that Parkinson's patients have altered levels of vitamin D. And people for a long time have been thinking, well, vitamin D is important. Maybe we need to raise the level of vitamin D. Now, maybe you do. I can't say definitively that you don't. But what we put forward was, in fact, that this part of it is happening without doubt. These genetic variants are changing vitamin D levels. But what they're also doing is in the brain, they are affecting this pathway, which is NAD. It's this generation of this energy source in the brain. That affects cognitive activities and abilities and the development of those problems that we see in Parkinson's. And really notably, there's a trial which is running overseas now where NAR, or it's a biological form or a form that you can feed people of NAD, is actually being used in a clinical trial and has been shown in the phase 1 to have quite a significant effect, so much so that they're moving that through to phase 2 and hopefully into phase 3 trials, right? And it's alleviating some of the cognitive issues that are associated with some forms of Parkinson's. And so this is real important, right? This sort of information here is linking the genetics through to things that might actually make a difference. But one of the things that we noticed when we ran our Mendelian randomization is, if you remember, I talked about that protein called alpha-synuclein. And alpha-synuclein has been associated with Parkinson's for a long time, right? It forms the plaques. Those plaques seem to be associated with the destruction of the dopaminergic neurons in the nerves and the tissues in the brain. And so that's really important. Now, when we ran our Mendelian randomization, we noticed something very interesting about alpha-synuclein. Now, I'm sorry, I've got this graph going up instead of across as I had the first one, but anything below the line here means that it's protective. Okay, this is the risk that a normal person has. Here, if you have this particular variant or switch controlling synuclein, then synuclein is protective. Okay, and it runs down here, and that's a 75% protection compared to controls, right? It's a big protection. But I just told you that synuclein is actually pathogenic. Well, if you look here in this tissue, and this is the skin, we have a different control element or a different switch controlling synuclein, and synuclein is actually risk. It's about 2.4, which means the risk for an individual has gone up 1.5 times what a control person would be. Now, that's a big increase in risk, right? 150%. Big increase in risk. So how is that possible? Higher risk, lower risk. How could one gene do this? how can it be both risk and protection? You know, when we first saw this, we thought, oh, we've done something wrong, right? You know, that's what you think immediately, right? So we checked. We don't think we have done something wrong. And this is all very new, so we haven't published this yet, so I'm telling you guys first. So I hope it makes sense. So a gene, as I described it, encodes a protein. They encode these enzymes and the structural things that make up our cells, that do all the functions that we have, right? And that's the way we think about genes. And that's true, except when it encodes multiple proteins, okay? So the original definitions of genes was one gene, one protein, right? And that's the way that often people talk about it. And it's the way that we talk about it if we're a little lazy in what we're saying. But the reality is that our genes are made up of component parts, and they actually encode multiple proteins. So when a gene is read, it's made into this messenger molecule, which is called messenger RNA, and it consists of exons, which actually make the protein or encode the protein part or the information, and then it has these other bits which are not containing information that's important for the protein. What happens as part of the expression process of making a protein is that this pre-RNA here is spliced so that the red box joins directly to the yellow box, joins directly to the blue box. That then is translated and made into a protein, okay? Alternatively, you can splice this a different way. So the red one joins the green one, which joins the blue one, which comes to this, which then makes this particular protein, a very different protein, but from the same gene. And we think this is what's important with synuclein. So, synuclein itself, which is the SNC or the SNCA gene, which encodes alpha-synuclein, okay, it makes multiple proteins. And all you need to know is this. The full-length alpha-synuclein is about 140 amino acids in length, okay? And it consists of a piece, we just call them pieces, you know, there's an internal piece and there's a middle piece and there's a C-terminal piece, right? Because we like to do that, but it just has three basic domains, okay? Internal, middle, and C-terminal. There are a couple of other versions of synuclein that are made at the same time. One is 66 amino acids long. That's just this part here, basically. One's 112, which goes right through to here, okay? Those proteins are really important. They're really important because experiments in vitro have shown that, in fact, if you take the 112 and you mix it with the 140 amino acid version of synuclein, you make aggregates, and you make them really fast, okay? The 66, by contrast, if you mix that in, you slow down the formation of aggregates. You slow it down. So let me show you how this works, okay? And to do this, I'm going to come back to that little model that I showed you at the beginning, okay? So let's think of the 66 amino acid protein as the loop bit of our Velcro, okay? Nice, short, okay? Our full alpha-synuclein is this. It's got a nice loopy bit of Velcro, and it's got a little ball here with the hooks on it, right? The hooky Velcro. Now, the hooky Velcro is our 112, okay? Now, synuclein naturally will form aggregates, okay? But it does it pretty poorly, you know, pretty slowly, because you've got to find a tail's got to interact with a hooky bit, and that takes time, and that's slow, okay? If what we do is we chuck in more hooks, okay, then what happens is the hooks will connect our tails and it out real quickly. And in fact, you can connect all three of them to the same hooky bit, right? So now I've got three forms of alpha-synuclein that have all aggregated. But then, of course, they will all stick because they're in close proximity. They all stick to each other's Velcro bits, and I've got a real mess of a protein, right? Three different ones all stuck together. This is an aggregate. Not only does that capture synuclein, but it'll capture other stuff that's floating through too. Other proteins like my pen, right? Gets caught up in this, gets mixed into the mass, and that's a problem. It's a real problem, and that's the problem that seems to be associated with the destruction of these nerds. Now, if by contrast, I have my proteins here, and I chuck in my loops, my 66 amino acid, what that does is it binds and it actually covers the sticky bits, the hooks, which means that when I get my other one and they come together, well, they don't stick. Oops. They don't stick, right? So it slows down the aggregation process because it blocks the sites that actually allow them to stick together, okay? And that's the key. That's what we think is the key. So if we look at what we found for synuclein, it's really quite simple, okay? This graph isn't simple, but I'll explain it. It's simple, okay? What we have is in the blood, we express a whole lot of forms of synuclein. There are two that you need to look at here, right? One is this one here with the dots in the green, which is this one, okay? The other one is the one here with the circle, which is this one, okay? This TPM just means the number of transcripts we find per million, the number of copies of this we find expressed in a cell per million. Now, in the blood, you see, we see some of this green one. That's cool. We see more of it here in the skin. But critically, what we also see is that 66 amino acid version that I just told you about starts out good in the blood and drops off to nothing in the skin. Now, what does that mean? What that means is that the skin is where we see risk. What we're doing in the skin is we're not making the loop version, which means we're not making the one that can block the activity of these, but we're increasing the ones that have the hooks. So we're making more of the ones with the hooks. And because we don't have the ones that don't have the blocking power, we're actually speeding up the whole process of making these aggregates. And that, in turn, increases your risk over a lifetime of developing Parkinson's. That's the theory. That's what the information says. Does it prove, or is there evidence for it actually in patients? So in patients, there is evidence for this. So Parkinson's patients, who we can take Parkinson's patients, there's a very, very nice study, which is called the PPMI study, which has been run in the US, sorry, through Michael J. Fox. And that study, basically, they genotyped patients. They did what I talked about. They looked at their genetic information, and we can look at those patients. And if we look at those patients, what we see is there's a subset of patients who actually show exactly what we're predicting. They have an increase in the 112, that is the hooks, and they have a decrease in the 66, which is the loops. And this, we think, is affecting the aggregation of alpha-synuclein in these individuals. Now, we can go beyond this. This is genetic, this study here, right? This is looking at the genes, the transcripts. We can actually go beyond that, and we can ask, does it actually work in their cerebrospinal fluid? And there are assays that have been developed, really beautiful assays that look at the speed of aggregation of alpha-synuclein. And these assays are being used now as part of a diagnostic in some places. And what they do is you put a sample into a tube, you add in some bits and pieces, and it shows you how fast the alpha-synuclein will actually aggregate. And that is forming a part of the diagnosis for Parkinson's in some places. Now, these were run initially on the PPMI. And if we look in the PPMI and those patients that I just showed you the genetic information for, what you actually see is that those people that have the right genetics that we think are doing this actually show a significant difference. The bulk of them, 77% of those people who have this, it's a very small sample number, and I apologize for that, but this is the most we can get at the moment. But those people, 77% of them are actually showing fast aggregation of synuclein compared to only 3 who don't. Okay, so 77 to 20%. Whereas in the rest of the population, the rate is around 64% and 22%. And this is a big difference, okay? It's not statistically significant here because of the numbers that we're talking about, but this is a difference, okay? And it's very important. Now, it's important to note here that also there's a number of patients here who are still showing fast aggregation. We think that might be due to other things or maybe environmental signals, but that's a different mechanism. The mechanism we're talking about is specific to people who have this particular genotype, what we call GGGG. Has no meaning whatsoever, but it's a shorthand, I'm sorry. Okay, why is that? This is it. Our results support a model for Parkinson risk, which is basically that the synuclein protein, as I described here, this thing basically aggregates and it forms an aggregation, okay? And it does that slowly. But it's 2 stages. One stage is the nucleation of the aggregate, so making an initial aggregate, and then you can basically take an initial aggregate, break it up, and make additional aggregates, okay, and that speeds the process up. The 66 amino acid version of this, which is genetically controlled, stops this process or slows it down. The 112 amino acid version, which is again genetically controlled and is something that is inherited from your rates of production of the 66 and the 112 are inherited from your parents, those things, this 112 speeds up this process, which speeds up formation of these nasty aggregates. That's part of Parkinson's for a subset of people, not for everybody. It depends on the genetics that you inherited from your parents. And that is important. It's important because now, and in fact, yesterday, there was a paper that came out, which was mentioned, actually, no paper, was mentioned in the New Scientist. But in the New Scientist, they talked about this, and they were talking about protein aggregates, in particular synuclein aggregates. This is their representation of a synuclein aggregate. And they were using an antibody drug called, I can never say these drugs properly, I'm sorry, it's prasinezumab. Terrible name. The monoclonal antibody, okay? And it's targeting alpha-synuclein in these aggregates. And what it's doing is it's slowing the progression. Okay, antibody infusions seem to be slowing the progression of Parkinson's. Now, that's using the antibody to target the end product. My question to you is, if we're right, and it's the genetics that we're talking about that we inherit from our mum and dad that set the rate of this aggregation over your lifetime, what if we actually interfered with this ratio, raised this ratio here, the amount of this one in people who had lower levels of it? Would that slow the progression and remove the need to do this and remove the risk of developing the pathology that ultimately leads to Parkinson's disease? Thank you very much. I'll just give you some acknowledgements. Obviously, I work at the University of Auckland. I've got to acknowledge most of this work has been done. The work I've talked about today is by Srimal, but other people have contributed a lot of work into this. Katrina, Oyedele's a new student with us, William, Eugenia, Teaza, Srimal, and Sophie as well. We've been funded by the Michael J. Fox Foundation, the Silverstein Foundation, Dine's Family Trust. Anthony's obviously at the University of at Garvan Institute in Sydney, and Ralph has also helped us with a bit of work on Parkinson's, and he's at the ZMBH, which is in Heidelberg in Germany, a former student of mine. Thank you very much, and look, I'll take any questions that you have. I notice there's only one in the book, but that's cool. Hopefully there's more. Thank you.
Well, thank you, Justin. I think the first comment I'd like to make is I'm so pleased you're on our team, because that is some amazingly complex things that you were talking about, but you have a remarkable ability to bring it down into a way that we can understand with your different demonstrations. So thank you very much.
Thank you.
Yes, the first one that we have is about that unpronounceable drug that you brought in your last slide.
The monoclonal.
The question is, it seems to support your approach because it failed in an earlier drug trial when results weren't significant across the whole cohort.
Yeah.
The second interesting point was it appears it took a year for the drug to show a slowdown in the motor deterioration in the subgroup. Yeah. What are your thoughts regarding why it might take such a long time to see results? If we are looking for regeneration of neurons, and given PD progress is slow degeneration, should we expect it to equally take time? How will a long time to respond be allowed for in your drug trials?
Okay, so drug trials like that. I mean, this is one of the big issues that we have. So one of the things that we've been trying to think about is how we can stratify or use the information that we've got to stratify people going to trials. Now, what we think is that people have different forms of Parkinson's. And so we have actually done some retrospective analyses with Anthony, he's led those, of some trials where they had genetic information. And we've actually shown, or he's shown very clearly that we can stratify people using some of the information that we have. And actually show that for a subset of people, other drugs have actually worked. But when we put them across a whole cohort of people, given that everybody has a slightly different form of Parkinson's and the genetic contributions are different and individual specific, you lose that information. Okay, so it looks like the drug doesn't work across the whole cohort. And that's because it doesn't. Any drug that we give people, 70% of the people we give it to, it's not going to work. A subset of people, it will. Same thing for Parkinson's. But the information that we're starting to get now allows us to start to think about stratifying people and actually making sure that we're using the right people in the right trials, or at least we can confirm that we're using the right people in the right trials and see the effects of these drugs in the right people. I think that will ultimately speed up drug trials. Things like the monoclonal antibody drug that we're talking about here, time for those effects is difficult because obviously when they're given to people, they're given late. And so they try to design these trials to see the impact over a period of time. And there's a set period of time where they try to do that. And they have very specific measures. I don't know what the measures are for that trial, but they are usually set quite stringently. And that's all to do with the number of people that you treat and the amount of variation that you expect normally and what you would expect to see over a period of time. Given treatment, if that was working. So there are statistical ways of doing that are all done by the trialists. I don't know the exact details for that one, but it is very, very carefully controlled. Yeah. Does that answer that question?
I think so, yes. Thank you, Justin. So someone else is saying, could you make just brief comment around the environmental measures? And that ties in with one of my questions. So let's just say it is Roundup, for argument's sake. So, and I spray Roundup liberally and I get Parkinson's. Is that it now? Do I have that? Can I spray liberally with Roundup and it not make me any worse? Or does it continue to affect me even after I've got Parkinson's? Assuming it is the causal factor.
Yeah, okay. So will it continue to make it worse? Environmental factors are, we haven't moved into studying environmental factors yet fully. My focus has been around the genetics. The genetics do interact with the environment. That's absolutely clear. What the factors are in the environment that are affecting it, I don't know what they are. There has been studies and there have been things that talk about pesticides and other things that are in our environment and their potential role in impacting on Parkinson's. I think once you start down the path where you're seeding these types of aggregates, right? These aggregates behave like a prion. So what they do is they start to form themselves. They split, they keep grabbing other stuff and it makes more and more. And whilst the aggregates themselves, we don't think the aggregates are transmissible from person to person. Okay, so it's very important to stress we don't think that. But within an individual, if they split, then they may well, they'll continue to build up. So at that point, continued exposure to something would probably not be advised, if that was the trigger. But I think the process is underway already. And risk, your genetic risk has been affected, and that I think is already underway.
So how certain are we that the alpha-synuclein is causative and not associative?
I think so. Our indications are that it's causative. The evidence that's there indicates that it is causative. These assays that they're running, the fact that we see these increased rates of alpha-synuclein aggregation in Parkinson's patients, I think that is indicating causation. Guys like me are always very reluctant to say definitively that it is causative, because someone will always come back at me and say, well, you haven't proven that absolutely definitively. The statistical methods indicate it is. The biological evidence indicates it is. I think it is. And I think there's more to that story that I didn't show you, which links in other aspects of the pathway of processing of alpha-synuclein as well, which come through as being causally related to Parkinson's. So I really do think it is causally related. Yeah.
Right. So we get asked the question, especially someone in my role, but our educators get asked the question, so is Parkinson's genetic? And I think we generally tend to I give an answer that goes along the lines of, we think it's maybe 10% of it is genetic, but there has to be something else and something else, and we don't know what the something else and something else is.
Yeah.
Is that, in terms of layman's terms, as the takeaway from today, is that accurate enough?
So I think, the evidence from NALS is that it's up to 36% of heritability, right? So 10% is a bit low, I think. I think it is, and I think there's strong evidence that there is a genetic component to Parkinson's disease, expanding up to sort of 36% of the heritability. Obviously, it's only risk. right? We don't think in deterministic ways, because
Yes.
you can have, people, there are adults walking around who have a mutation in the cystic fibrosis gene, a delta 508, and they don't present with Parkinson's. They don't present with cystic fibrosis, right? Because there are other things that can still modify that. So it's never 100%. There's always something that can modify. But the reality is that there's a strong genetic component to Parkinson's that interacts with your environment, as does everything. And that is, I think, the reality here. Yeah.
Right. So that's not unique to Parkinson's, as you were saying at the beginning. It's just the way life is for us as humans.
It's just the way it is, right? I mean, we all have genetic variation, I mean, we sequence children in acute care at the moment, and their parents, and we see the variation between the children and their parents, and we can link that through to the phenotypes that the children are demonstrating in the hospital, and the clinicians can use that as part of the information that they are using in their decision making about how the treatment goes for those children. We're doing that now. I mean, the reality of this is that genetics set your risk for different things. Yes, the environment's going to modify that. That's fine, but it is set. We do have, for a large number of things, conditions where it's to do with, our cell behaviors and those types of things. There's a strong genetic component to that. Absolutely.
So someone's asking about the vitamin D comment that you made. Should we take a chance at it and just load up on vitamin D?
Should you take a chance at it? Like, if it makes you feel better, there's and we get asked these questions, I guess occasionally. I'm not a clinician, so I do not give clinical advice. But I think, there's well-known placebo effects for things, and I can't say that taking vitamin D is not going to help you, and I can't say that it will help you, but I think if you take it and you feel like it helps you, then that's a good thing.
Right. And maybe the last, because we're out of time, but just the last question is quite a complicated one, so I'll read it out. I've been told that I have Parkinson's caused by a recessive mutation of the PARK2 gene that I have. I was told that this is the most common cause of inherited Parkinson's, and also that it's extremely unlikely I would pass it on to my children. Do you know anything about PARK2 and its cause? And is it causal with 2 copies? Or would environment play a role?
Okay, so the key thing here is that what you've been told is you've got a recessive mutation, within PARK2. Recessive means that in that graph that I showed you before, there's the big B, the little b, right? The recessive one is the little b. And what that means is if you have big B, then big B dominates, right? And it's a function. Now, if you've got 2 copies of a recessive, then what has to happen is, in order for your children or anybody subsequent to you to actually get the same thing, they have to have 2 copies of the little b. And if you go back to that graph, that means that the other person, your partner, must at least have a big B and a little b. And then if they have a little b, by recombining everything, it's only 25% of the time that you'll get 2 little b's. Okay, so that's all to do with that, genetic gamble that we take when we're born. And but as I think you've been advised, it's unlikely that would happen, unless your partner has big B little b situation, so has a recessive PARK2 as well.
Thank you, Justin. On that note, I think we'll bring it to a close. Thank you so much for your time and your expertise in bringing the complex to us. And obviously, it's something that does trouble us. Are we going to see a cure? Is what we all ask ourselves the whole time.
Yeah.
And are gene type therapies going to come our way in the near future? And so we're just so grateful that this work is being done. And it might not be in our lifetime, but certainly for the following generations, they will be better off because of the work that you're doing. And so we're very grateful for you and your whole team that you mentioned.
Just before you go, there's one question here. I think Robin Cotton's put a question about how realistic is it that Parkinson's patients will be able to get genes typed to enable targeted therapies. So my answer to that one is we are currently doing sequencing now, and we're trying to expand that sequencing to other things. So we will try and get that sequencing for Parkinson's people, because I think it is very important. We just have to get the funding to do that, but that's something that we are really actually trying to do, is to get that. We did do a proposal some time ago about trying to individualize this way using the information we have. But I think we're trying to recycle that and fix it, and hopefully we will be able to make that happen. But it's delayed. That's all.
Right. Well, thank you for keeping on it. So thank
Right.
you everyone for your attendance. If you would like to watch it again, you can. Justin's going to be on again at 7 o'clock tonight and repeating the same presentation. So you just have to re-register, because the link that you've been sent for the 11 a.m. session will not work for the 7 p.m. session. Alternatively, it is being recorded, and you will be sent a copy once it's uploaded to our YouTube channel. You will get a link to that in the next 5 days or so. So it was a lot to take in. So take the opportunity to have a watch again, and to learn more. So again, thank you, Justin, to you for your time. Thank you to everyone for participating, and thank you for the progress that we're making on this journey called Parkinson's.
Thanks.
See you later. Thank you so much. Bye-bye now. Bye, everyone.